scientists! Thanks for joining me again today for another fun science experiment. Today we're doing one of my all-time favorite science experiments using cabbage indicator. So we will boil some cabbage, take out all the anthocyanins that make it purple, and we're going to use that as a pH indicator to test whether things are acids or bases. It's going to be super fun, so let's go into the kitchen and get started.
To make our cabbage indicator, you're going to need to chop and boil a head of red cabbage, and then you can strain the cabbage out so that you have just the liquid. This liquid has anthocyanin in it, which is a chemical that makes the plant look purple. This is the chemical structure for the molecule anthocyanin, which is what we're going to be using as our indicator in this experiment. And you don't need to know or understand anything about this picture except that it is an indicator because it has these two reactive groups on it that are highlighted in yellow that will interact with acids and bases differently and change color so that we can see if something is an acid or base. Anthocyanin is found in many plants like red cabbage, butterfly pea flowers, blueberries, hydrangeas, and beets. It's a very common color in the plant world. And when it is exposed to an acid where there are extra hydronium ions, the hydrogens from the acid will bond with this extra reactive oxygen on the anthocyanin, and that will turn the solution pink. Whereas in a base, an extra hydrogen on the other reactive group of the anthocyanin will leave the anthocyanin molecule to create water, and then it will also reflect light differently, but it will look blue. And that's what makes anthocyanin such a great pH indicator, because it looks vastly different in different pH levels from pink to purple to blue, and it's reversible. Now, when the anthocyanin is exposed to an acid, like this vinegar here, it's going to turn pink. It changes the chemical composition of the anthocyanin molecule so that it reflects light differently. Similarly, if you add a base like baking soda, it's going to turn blue. And that's because another chemical change alters the anthocyanin molecule so that it reflects light differently and it looks blue. My favorite way to do this experiment is actually using the cell culture plates that you can get from scientific supply stores. These ones actually came from my lab because the plastic had expired. But it's a really nice way to do the experiment because it doesn't require a lot of liquid. You can actually aliquot it out ahead of time, so the kids just have to add their acids and bases to it. And they can see the different hues of purples and pinks and blues next to each other, which is really nice. We like to also use litmus paper to compare how the pH looks on litmus paper versus in our cabbage indicator. You could also use test tubes to do this experiment, which you can find a little bit more easily online, or you can just use plastic cups that you get at any store. These work perfectly well. It's a little bit more difficult to prepare everything ahead of time, but no matter what you do, kids are going to love this experiment. It's so fun to see the colors change right before your eyes, and I hope you really get to try it with them because it's super fun.
Thanks for joining Simon and I today as we made some cabbage indicator and tested a whole bunch of solutions to see if they were acids or bases. I hope that you learned something and that you had a lot of fun doing this experiment. And until next time, keep discovering.